We heard at the talks preceding this, multiplexing for ion analysis and this mastering. And what I will discuss now, this focus, will be multiplexing ES orthogonal domain. Great, yeah. So very excited to introduce this for the first time publicly. I think this is a really interesting technology that we've been to scale throughput for analysis. So why would we want to increase the throughput of trivial from analysis? Today, the reason would be to generate large-scale data, such as maybe analyze large cardboard analysis. There's many covariates that come along with analyzing pathologies that you would want to investigate the results and the causes of the covariation of status, sex, of age. So like this is pathologies. Like generating large-scale data, you might be able to investigate this. So if we had this goal of analyzing 1 million samples, this really ambitious goal, by today's standards of what most people would consider high throughput proteomics, it would be maybe 100 samples per day. And if we wanted to analyze this, it would take many decades, cost many tens of millions of dollars to do this. So technically, very impractical and infeasible. So there is this really strong urgent need to increase the throughput of proteomic analysis. And so how can we do this? We can do this really simply by just increasing the rate of data acquisition. We can churn through samples a lot faster. So we can shorten LC separations, for example. This is work done by SNRL from the Wamsley lab, where they use an EvoSep system to get down to less than a minute-long readings. So they're getting really fast separations to enable more runs per unit time. This does come with the downside that people are mostly familiar with. As you reduce your separations, you're going to likewise reduce your coverage per unit time. And so there's other ways that you might want to go about multiplexing or go about increasing throughput. Another way would be to just make more efficient use of the time that you spend analyzing samples. This is worked by Shunol from the Ryan Kelly lab, where they have 2 columns. One is undergoing all the overheads associated with sample loading, column equilibration, and the other is actively separating chemical analytes. And so you can put all your overheads on one sample on one column, while another column is actually doing the things that you want it to do. And you can do this switching back and forth, and that makes more efficient use of this technology. Another way with the multiplexing samples. So these approaches are all complementary. You can do both or one. Today, the talk is going to focus on multiplexing. And a common way of achieving this would be to do isobaric labeling, in which case you'll have maybe you'll use TMT and you'll have a 35-plex, for example. These are going to be, once you build them together, you're going to have like this superposition that's observable at the NS1 level. You can fragment these charged peptides and you'll get sequence information. And you'll get quantitation that is specific to each individual sample. This has really high sample multiplexing capacity with a 35-plex, but the coverage that you get per unit time has a ceiling on the number of down-2 scans that you can possibly take. So we can really scale essentially the sample throughput, the coverage that you get per unit time is going to come at a detriment. Another technology that's been growing in popularity a lot over the last 5-10 years or so is data-independent acquisition. This is a way of acquiring data that will intentionally generate chimeric spectra, so they could quantify dozens or hundreds of peptides in a single scan. And this allows you to get really deep proteome coverage very quickly. And so a few years ago, we developed this capacity to enable multiplex data-independent acquisition so that you can get deep proteomic coverage while also having this multiplexing effect. And the benefit of this is that you'll increase your throughput multiplicatively. And then as a result of this, because all the costs are associated with LCMS time, by increasing throughput, you also reduce the costs. So you get this dual benefit effect. Now, what I'm really excited to talk about today is this new capacity of multiplexing samples in this orthogonal dimension. So rather than multiplexing samples in this mass domain, potentially we can multiplex samples with time offsets. So we could temporarily encode samples to undergo the exact same separations and just stagger them at time. And so in this way, the time offsets would be orthogonal, would basically be orthogonal to mass offsets. To give you a more intuitive view of how this might look, we can start with an example that I think most of us maybe are familiar with, where you have samples that you're going to load onto a column. Your peptides will bind this column, and you'll flow increasing amounts of organic solvent over that column, and you'll have peptides that elute in a way that is highly predictable and repeatable. And so this is being illustrated here with peptide I, that's red, followed by yellow, green, and so on. And the idea with timeplex is that if we stagger in time the introduction of this gradient, then all of these peptides are going to be separated by this systematic time offset that is going to be held constant throughout all these peptides. And so now, rather than encoding samples in the mass domain, they're encoded by time separations that are systematically held. They're going to undergo the exact same separations. In this way, we can encode this time differential by just having transfer lines that contain different volumes. As you flow at a given flow rate, they're going to have trapped times that are effectively different, and that's how you can code very simple this time differential. And potentially you can do this for as many time plexes as is practically feasible. And today I'll illustrate a threeplex. Now what's really, I think, very cool about this approach is that it's orthogonal to the mass dimension, which actually means that you can do both. You can multiplex in this mass dimension with plex DIA, and you can multiplex in the time dimension with timeplex. And so as effectively you have a 3-plex DIA, and you have a 3-timeplex that effectively gives you a 9-plex. So you can actually quadratically scale the throughput that you get and also the cost savings. So any developments that we make on plex DI side with developing PSM tag, 9-plex, and in the future, it's all going to contribute synergistically with timeplex, and we'll get this really rapid accelerated increase in throughput and cost savings. And so I was really fortunate to have Kevin talk before me. So how to go about actually analyzing this data, and it ends up being very similar to exactly what Kevin had said. The only difference now is that we're going to need defined retention times, expectations of when a peptide is going to elute for a given sample. As long as we can have that, then we're going to have sample specificity for each peptide. We're going to know each peptide originated from a given sample, and we can, when we do the search, we can basically just set thresholds on when we'll consider a peptide to map to a given sample. In which case, what I'm showing here is real data. This is the result of a fine-tuning that we have done and integrated automatically into JMod, where I'm highlighting in magenta color a particular charged peptide, a precursor. The data doesn't always look like that. You might have a library. Library is basically just an expectation of when a peptide is going to elute. Maybe your expectations for when a peptide is going to elute are really off, like very wrong. In which case, we're going to have a really hard time figuring out what peptide originated from what sample. This is actually a solvable problem. This is not something a result of timeplex not necessarily working. All of the error that we're seeing, all the jitter we're seeing, is a result of the library retention time not accurately reflecting the separation that these actually have. And so we did a lot of work on implementing a flexible solution to solving this problem, which is getting accurate retention times for peptide elutions. And really happy to discuss this more in detail. For the sip time, I'll keep it brief and just say basically we've trained a model. We have our own custom predictor for retention times that we then use transfer learning on based on a subset of initial identifications that we get. And we adjust all of our expectations in this library now. So we can go basically from this left-hand side panel to the right-hand side panel where we have really reverse expectations, strong expectations for when peptides are elute. We can get basically completely unambiguous identifications for which peptides belong to it. And happy to answer more questions in more detail about how we actually do that in Kubernetes. So how well does this actually work? We have this way, potentially, of analyzing these samples. We wanted to benchmark, are we able to get sample specificity with time-plex? So to do this, we went to our trusty multi-mixed species benchmarks. We have time-plexes that are offset like 4 minutes in this case. All these samples, all 3 of these samples are going to have human. And then the last 2 samples are going to have yeast. So it's going to be missing yeast in first sample, and then we have this plant proteome. It's not going to be present in any of these. And we're going to search this data with a spectral library, FASTOS, that is the map to human, yeast, and Arabidopsis. And what we should see is that we have good human identifications across all the different samples. We lack yeast identifications in the first sample. We don't have any Arabidopsis identifications in our NA for samples. And so here what I'm showing is the raw data from this time-plex run. And so what I'm showing here at the bottom is the total extracted ion chromatogram. And we have GAPTH, a precursor that corresponds to GAPTH, and plotting intensities for precursor for each one of the 3 time channels. So we have good appreciable signal in all 3 of the time channels. For the human and for the yeast, there's this absence of signal in our first time channel, suggesting that we're getting good ADR at least for these 2 peptides. So how does it look for the rest of the proteome? So if we do a more systematic analysis, we're getting about 20,000 or so identifications of human, a large number of identifications for human in all of the time channels. And I'm plotting here the intensities on the x-axis. And we have very low identifications for yeast in the first time channel as expected, and very low identifications for Arabidopsis in all the time channels as well, suggesting that we had good false discovery rate control. Okay, so to summarize this analysis, we have this potential to multiplex samples in this time domain that's orthogonal to mass. And its time offset is really analogous to mass offsets. So time offsets and time-plex are analogous to mass offsets applies to IA. We can leverage this almost as equally well as you can in time-plex, but the requirement to do that is that you have accurate retention time predictions so that you can get sample specificity. And so we showed that we have this approach of searching this data that can give us good control of false discovery rates, shown here. And so now we want to benchmark the performance of time-plex relative to more established methods like layer-free DIA and plus DIA. And we've also done this combinatorial time-plex and less DIA. We want to see how well this nets out to these established methods. So to do this, we had a sample, which we're going to call A, B, and C. They had 8 nanograms of human protein lysate in all 3 of the samples, and then a variable spike in of yeast so that we can benchmark for quantitative accuracy. And to show the generality of this implementation, time-plex can basically be implemented in many different instantiations. We have 2 different implementations that I will show today. The first of which, you have 3 combs. They're going to be loaded, and then as the solvent flows over them, they're going to be joined and then spray from a single emitter tip. The other one is going to have separate emitters throughout using ion optics columns. So 2 different implementations. So what do we see when we get this data? The current implementations are good proof of principle. They work reasonably well. What we do observe is that there is this drop in intensity, which is likely the result of increased ion plume density as you spray multiplicatively greater solvents at the mass spectrometer. There's going to need reduced desolidation, which likely is the result of what we see here, where we have basically 1 sample that's loaded, spraying at the mass spectrometer with the 1 column, or another sample that's loaded, spraying at the mass spectrometer with 2 columns that are background spraying. So we have about 2-fold drop in intensities with this current implementation, and about a 20% drop in protein identifications. This drop is probably going to be less significant as you go to higher and higher protein amounts, because 2-fold drop that nanograms on Explorers 248 actually ends up big very easy. Yeah, versus like injecting 10 times that. No. So this is the implementation we're going to use. We think that it can definitely improve in the future. And we want to investigate how old does the data type itself stack up. So these other implementations can, assuming that ourselves or others in community are going to approve this implementation, how old is the data type actually look and stack up? So we're going to control for the implementation so we can get a look at what the data actually looks like. So what I'm showing here are the number of proteins that are sample specific that are identified per run. And so what we see here is that we're getting about almost close to a threefold scaling of plexi and the label free DI. So we're getting proportional scaling. So we're getting about threefold more proteins per run that are sample specific for the true plexes. And then for the combinatorial 3 plexi, 3 time plex, we're getting about 6x the throughput, give or take, in terms of throughput defined as number of proteins identified pre-time. And so, yeah, we're getting proportionally about 2/3 coverage, which we think that, and I'll present at the end, we have some evidence to suggest that this can be improved many different ways. Well, one of which is analytically with spectral libraries, which Kevin hinted at early. We have really similar trends in the other implementation that we have. So this is the single emitter join flow, very similar trends for both these. So the next thing we did was we looked at the quantitative accuracy of this time plex. And first thing that struck us is that there is these changes in relative ion intensities as a function of the retention time. This is common if you have samples that are running on multiple different columns. And for example, maybe you need to maybe one on this batch effect, and you need to basically correct for this. This is something that most search engines will already handle, is this normalization as a function of retention time. We're able to, so basically what it is, we normalize for these relative intensities as a function of the retention time, just fit a lowest regression and fit that basically to 0. And then that basically solves a lot of our issue with quantitation. And so now we're able to basically get quantitative accuracy that reflects the mixing ratios that we would have expected from this spike count. Okay, so this is showing the quantitative accuracy for the separate emitters approach and also for the single emitter approach, suggesting both implementations give quantitative accuracy. All right, so now we're going to compare the different methods, the label-free no-plex and plexi to these other methods. So these are the established methods that we have. Plexi is going to increase sample throughput threefold multiplicatively. We observe that we have very good quantitative accuracy, even by multiplexing threefold. This is what we observed for a few years ago as well. And with time collects, we're also getting this scaling of sample throughput by threefold and maintaining good quantitative accuracy. Now, if we go to the combinatorial 3 plexi, 3 time plex, the median accuracy remains the same, albeit this spread does increase. And we are increasing sample throughput by 9-fold in this case. So to summarize the benchmarking, timeplex can be implemented in many different ways, some of which we are hopeful that will improve in the very near future. We're getting this about 2/3 proportional scaling of covers. We think that we can improve this using improved spectral libraries, which we'll show data for towards the end of the presentation. And we're observing good quantitative accuracy with timeplex and also with this combinatorial timeplex plexi. Okay, so next we wanted to do was apply this combinatorial approach to analyze single cells. And the reason being is that if you wanted to enable single cell analyses, there's many different analyses that you might want to do, such as profiling, understanding what phenotypes are heterogeneous in the cell and seeing what proteins covary at abundance with that phenotype. And you can learn things about fundamental biological processes. To do that, you need ideally quantifying thousands, tens of thousands, hundreds of thousands, single cells. And so to do all that, you'll need to increase the throughput. And if you basically just rely on plexi or timeplex, it's going to give you this multiplicative effect. It's going to be very useful. But what would be even more useful is using this combined approach where you can really quadratically scale the throughput that you get by using both time-plex and plex-DI. So that's what we're going to demonstrate today. So we prepared single cells by NPOP. This is a workflow that was developed by Andrew Leduc and Nikolai Slavov's lab. And we're going to analyze K562 cells and U95% cells on a low-course instrument, the Explorers 240, just as a proof of principle to see how there is the quality of data that we can generate from these single cells. At the same time, we analyzed 100x diluted bulk. So we had these cell cultures that are going, and we took off some bulk sample and we diluted it to 100 cells worth. And what we want to do now is assess the quality of this data. So we can use that bulk diluted standard, the ratio of protein abundances in the K cells to the U cells as our value line. And we can see what did the averaged out single cells, what did their quantitation look like relative to it. And the idea being is that they should agree. We have strong correlation, 0.85, between these averaged out single cells and our bulk data suggesting we have good quantitative accuracy in our single cells. I'm highlighting two proteins, HMGA1, which is just a protein that's highly abundant, and FAVP5, which is a protein that is about 5-fold more abundant in U937 cells than K562 cells. And I wanted to look at what the raw intensities were in these single cells for these proteins. And so what I'm showing on the bottom right is the total line chromatogram for these single cells. And then I am extracting signal at the unismol level for these single cells. And what we see with the charged peptide, HMGA1, we have appreciable signal in all the single cells for this charged peptide. And there's no obvious differential abundance between them. But when we look at the charged peptide for FABE5, we have appreciable signal on all of them. And likewise, we observe this differential abundance even in our raw data, rather than just the averaged out single cells. So we also did some dimensionality reduction, and what we found is that, as expected, we're getting really clean separation between cell types, and that we projected also the bulk samples into the same space as their single cells, and they are co-clustered as well. And so I think this shows a couple of different things. The quantitation of the single cells quality seems to be good. And also the methods of acquiring the data, one with time-plex alone, and also this combinatorial time-plex plex-DI are giving very similar results, which I think gives us more confidence in this technology as well. So to wrap this up, we have developed this orthogonal way of multiplexing, which we think can scale quite well, especially when paired with time-plex. And proof of principle, I think we've demonstrated potential for an order of magnitude increase in throughput. And actually, if we scale this with the PSM tag that is in line-plex, then effectively we would have 27-plex. We can really rapidly accelerate the throughput of this and really reduce the costs and also the time for it to generate such data sets. Okay, so this is more so about future avenues, which I'll keep relatively brief. The reason I was saying that we think the coverage can improve is, to me, something that is really intuitive. As we're using this joint modeling approach to model spectra of peptides, we have complete reliance on the library that we're given. And so if that library is even slightly inaccurate from what we're actually observing, we could end up with some lackluster results. And it's not because the data is bad, it's because our model is ill-informed. So to assess the impact of this, I looked at two data sets that I had, plex-DI, which is 3-plex, and the 9-plex, which is the plex-DI time-plex. And I searched this data with two different methods. One was a library that was really well tuned to the mass tag that we used. So you can consider this an optimized library. And the other one was a library that was reflecting label-free spectra, so it's a bad representation of what the spectra actually look like. And if we searched it with the plex-DIA, there's really only a 5% increase in the number of precursors identified with the optimized versus the unoptimized. And potentially this is because there's relatively low overlap, which Kevin was discussing earlier. So it's not a huge impact. But actually, as you go to higher and higher plex, that spectral library that you have becomes increasingly important. And so this is something that we think that we can further improve the coverage to just very simply have spectral libraries that more closely reflect what is empirically observed at spectra. And so lastly, the future directions of Timeplex, the implementation that we have, we think it works pretty well. We think that it can be improved in the future, and we're excited about future developments and what other people will think is good approaches to move this forward as well. One of these that I think is particularly exciting and should be accessible even today is work that was done by Riggs McKinnon at Hughes Lab, which uses multi-segment capillary electrophoresis that temporarily encodes those samples for capillary electrophoresis and for metabolome analysis. And this should be usable on even today full proteomic analysis, as far as I know. Another approach would be running isocratic separations using size exclusion chromatography, which would allow for continuous gradient. Actually, effectively no gradient. It would just be the same solvent that's flowing through the same column, and you would sequentially just inject a new sample every X minutes, and potentially this would reduce overheads associated with that timeplex. So a lot of future work I think that I think is really promising. So I'd like to thank in particular the team that made this possible, everybody at PTI, all of our funders, in particular Kevin for implementing his timeplex module to JMA, and Peyton for doing single cell preps, and Nikolai and Harrison for guidance through this process, as well as I do want to say the preprint is coming along, should be pre-printed in the next couple of days or so, next week or so, and we will have all the data available on, all the code available on GitHub. Data will be open access on our website and elsewhere. And if you have any questions, you can contact me at this email here. And thank you all for your attention.
Thank you, Jason. That was great. I see we already have a question, but as I make my way over there, I'd like to start off with one. Is that we saw that our PSM tag had really good scalability going from single to 5 to 9-plex. Do you think the not quite linear scalability is intrinsic in any particular aspect, or do you expect improved results with something like our PSM tag?
Yeah, I think anything that is going to improve the spectral quality is going to improve this analysis and this combinatorial effect as well. So I would expect it to definitely improve things. The trick, I think, is, especially for timeplexing, getting really accurate retention time so you can have that sample specificity. And that's going to help things along as well.
Great talk. I wonder, maybe you've thought about this a little bit, but in the first talk, there was a bit of mention of how tags can substantially shift retention times. Have you thought of just maybe there's two totally kind of chemically different subsets of tags? One substantially delays retention times by, I don't know, 5 minutes or something, and then achieving a timeplex mode through something like that, where you can just mix all the samples together, inject them, and then they just elute differently, and this allows you to do timeplexing.
So effectively have like different mass tags that you would pool together. Yeah, so that's a really good point. I think that would enable a multiplicative increase in throughput, but because the tags wouldn't be on the same peptide, it wouldn't be combinatorial. And so this would actually enable, this timeplex modality would enable multiplexing in time, because you can basically, you can do exactly what you're saying and go maybe from like a 3-plex for with M-track to maybe you have a 9-plex PSM tag. You can combine them together, get a 12-plex. And then that wouldn't necessarily give you that combinatorial aspect, I think, if that answers your question.
So you need a different set of tags for each additional combinatorial.
Yeah, or you need to get, I think you would need to get the tags on to each peptide, right? You need to get a different tag onto each peptide to get that combinatorial effect, right?
Maybe not necessarily, or if you just had two totally, one tag that had 3 channels, and a totally different tag that had 3 channels. And so now you combine your 6 samples together, and tag one was 5 minutes later than the other 3 channels.
Oh, okay. So you're saying that they're like chemically
they're
the
totally
same?
chemically different, distinct tags.
Okay.
So you have like, you injected say M-track, you injected M-track labeled single cell in a sample with the PSM tag single cell labeled single cell. And say the PSM tag is more hydrophobic and generally elutes 2 2 minutes later per peptide. And now you have the PSM labeled and additional single cells for M-track, and you just totally treat them differently here. I guess there would be other challenges.
I think that would be, would that be an additive effect? I think it would, right? I don't know. We'll talk about it later. Thanks.
Hello. My question is a little bit more technical. You showed in a diagram that you're splitting the flow out and then going through 3 columns, and then you're either going into the mass spec via 3 sprays or basically backwards through your flow splitter into a single spray. How are you going to implement that as you go up through your multiples? Because it looks like you're probably, or it sounds like since you're going 927, you're just going to take that flow splitter, multiply it by going through another set of flow splitters and another set of flow splitters.
Yeah.
So I have 2 questions. One, how are you going to recombine them into a spray so that you're not just like blasting the front end of the mass spec with a bunch of liquid? And then also on the front end, you know, at what point do you think the LC pump is going to fail?
Yeah, great questions, great questions. No, yeah, we've had this exact same thought. And that's why we kind of went with 3, because it's the most usable. There actually potentially is a way to implement a 4-plex on this. But 3 is the most practical of this. Yeah, and like you're saying, potentially if we went to higher plex and we only have basically a cross, I think is what you're thinking about, where we have 3 inputs and 1 output. Yeah, if we wanted to scale this more, then you're right, we would need to have multiple of these crosses to basically join the flow. The way that we foresee this scaling in the very near future would be to have columns that hold very small volumes, so you can run very low flow rates without affecting your overheads. And then a result of that is that the cumulative flow that you get at the end of it would be hopefully not too bad. So that I think that in our mind is the way that we're going to go in the very near future. Thanks.
Related to that, if there's anything you wanted to expand on the challenges of implementing timeplex and how one might readily adopt it into their lab.
Yeah, thanks. Yeah, so in terms of that, I think that is very similar. So the way that this can be implemented, you could join flows. The thing that's really important when you join flows is that the cross that you use has a very small void volume, as small as you can possibly get it. I think Vicky Delco sells some crosses that are 50 micron bore size, and so it's a very small void volume that you would incur, because really you don't want to. You're separating all these chemicals on these columns that are really sophisticated. You don't want to basically create turbulent flow that's going to result in tailing peaks, and so just having connectors that are small void volumes so you don't disrupt the separations would be one implementation. There's also, you can use separate spraying approaches like we had demonstrated before. There are difficulties with that as well that make this practically a little bit harder. So I think the way forward probably will be doing joint flows.
I was wondering, how do you optimize the time delays?
Yeah, thank you. So yeah, thanks for the question. It's kind of interesting because it's very physically, the first assumption we make is that the columns are going to equally go back pressure, which for some columns that's definitely the case, for other columns it's not the case. And if you encode a difference in volume in those transfer lines, then theoretically if they hold the exact same back pressures, then you should be able to encode a very precise time offset. So like if one transfer line is 1 microliter and the other one's 200, and you flow, challenging my math, but anyway, you flow at a certain flow rate, then you're going to have an encoded time offset. And if the columns are relatively repeatable and robust, that back pressure shouldn't change, and so your time offsets shouldn't change too much. And that's basically what we've been observing too.
And a second question, so you mentioned library optimization at the end. What specifically about timeplex can you optimize for in a library?
Yeah, thanks for asking that. We can optimize, the most important thing is optimizing the retention time, so we can have good expectation of when peptides can researching this data using this joint modeling approach, having good expectations of how they're going to fragment is also really important. So it's a combination of both of those things. We can either, if we have an empirical library that has good retention times, basically you ran your sample, you searched it already, and it has retention times in your library that reflect what they actually should be, then you're pretty much set to go on the retention times. In cases where that isn't the case, we have built in a automated way of doing that fine-tuning in silico. And so even if you don't have optimized retention times, you can still get pretty usable data. Thanks.
Related to the retention times and the optimization of time-plex, how small can you get in your delta retention time? And do you expect that it's a limitation of hardware or computational?
Yeah, thanks. That is a more so a limitation on the analysis side of things. So if you can get your retention times to be incredibly accurate down to within 10 seconds, there's really no reason why you can't offset samples by 20 seconds, for example. And so you could go to really, really short time offsets if you have really high precision and accuracy on your expected retention times. So it's really just a function of how robust the system is, so that they don't overlap in terms of their time, in reality, and then also how well you can model the data. So I think in the future, definitely down the line, I think we can go down to less than a minute.
Any more questions from the audience? Check online. Nothing online. I'd like to remind our virtual participants to please do add questions to the chat. We are monitoring them. But if there are no more questions for Jason, let's give him another applause.